Your future is created by what you do today, not tomorrow. Hi everyone, welcome to Divya Zoo class. Today we are going to discuss about DNA cloning. What is cloning and how it is done? Cloning is the process of making a genetically identical copy of a gene or organism. Cloning is done using molecular biology techniques. First, the gene of interest is cut and pasted into a vector, such as plasmid, to form recombinant DNA. The recombinant DNA is then inserted into a host which make copies of gene. What are the three types of cloning? The three types of clonings are gene cloning, reproductive cloning, therapeutic cloning. Gene cloning, cloning of specific species of DNA. That means specific piece of DNA. And reproductive cloning means entire organism. Therapeutic, to treat diseases or replace damaged tissue. Some steps of cloning process here involved. The main steps, they are using two different kinds of enzymes. One is called cut enzyme, that is restriction enzyme. Another one is paste enzyme, that is DNA ligase. It is used to insert into a cell for replication. What is DNA cloning? DNA cloning is making an identical copy of DNA. It is commonly used in medical research both to create treatment and drugs as well as to research disease states. In a typical DNA cloning process, the gene or other target DNA fragment is first inserted in a circular piece of DNA known as plasmid. This is done using restriction enzyme, that is cut enzyme, and it is introduced or to involved in the recombinant DNA. That means it is modified into recombinant DNA. Next, the recombinant plasmid is introduced into the bacterial cell. The bacterial cell carrying the plasmid and selected grown up. As they reproduce, they replicate the plasmid and pass it on their offspring, making copy of the DNA that is called recombinant DNA. In some cases, we need several DNA copies to conduct experiment, that is, build new plasmid. Gene cloning methods, there are four major gene cloning techniques are there. Isolation of DNA to be cloned, insert of a foreign DNA fragment into a vector, transfer of our DNA into a bacterial cell, detection of recombinant clone. The cloning process, here several applications are used. For example, let's see how the DNA cloning is utilized to synthesize a protein, such as human insulin in bacteria. The basic gene cloning steps are cutting and pasting DNA. We know that what is cutting and pasting. By using restriction enzyme, they will be cut, and by using DNA ligase, they will be pasted. And next one, bacterial transformation and selection. Then the plasmid and other DNA fragments can be introduced into bacteria such as E. coli, used a process called transformation. And protein production. The bacteria serve as factories producing large amount of protein. Applications of gene cloning. There are many uses of gene cloning in the medical and research industries. For example, gene therapy and gene analysis. I hope that you can understand what is gene cloning and what are the steps involved in the process of gene cloning. Thank you.